Welcome back to Pathophysiology, students. I hope you're ready for your next lecture. We're going to go ahead and get started. So, pathophysiology, we are going to do part one of 2 lectures regarding the cell. So, which modules are we going to go through regarding the cell? We've got cell structure and function, cellular response and disease, role of genetics and disease, and neoplasia. So, this lecture is going to cover actually both of these lectures, the cellular structure, whoops, I misspelled that, function and adaptation, concepts of health and disease, cellular response, genetics, and neoplasia. So to get started, we are going to review some anatomy and physiology. So let's start off with the cell. We have 2 main different types of cells. We have cells with no nucleus, which are prokaryotic cells, which are plant cells, and bacteria have no nucleus, but he's a happy little dude. And then we have cells with a nucleus. Now these are more complex. So these are eukaryotic cells or human cells, and they can do complex things. So students, you have nucleuses in your cells. You can do complex things. So what is a nucleus? The nucleus holds DNA material. The nuclear envelope contains pores for material to pass to and from. Other parts of the cell, so things that you need to know that are inside of the cell. The mitochondria is the powerhouse of the cell. It makes adenosine triphosphate, or ATP, and it will be referred to as ATP throughout the rest of this lecture through oxidative phosphorylation, and it has its own DNA. So I want you to picture this. I'm really big on graphics and visualization because pathophysiology is tough content. It is a lot of hard memorization. ATP is the powerhouse of the cell. So I've made a little powerhouse here. It makes ATP. And do you see where I've put DNA? It's in pink. Mitochondrial DNA is only maternal. You only get your mitochondrial DNA from your mom. Okay, and this is what the mitochondria looks like in the cell. We have our rough endoplasmic reticulum. It has ribosomes. So we've got this rough guy. He is our rough endoplasmic reticulum. He's got our ribosomes on top of there. And look what he's making, students. Ribosomes make protein chains. Okay, ribosomes make protein chains on our rough endoplasmic reticulum. Smooth endoplasmic reticulum has to do with detoxification and lipid synthesis. So smooth endoplasmic reticulum, lipid synthesis, and detoxification. The Golgi apparatus is the distribution center. It modifies packages and ships and receives proteins from the endoplasm. So think of your Golgi apparatus as kind of like a distribution center or your post office. It packages things up and ships proteins to where they need to go. So this will be a good visual for your Golgi apparatus. When you're reading about the cell throughout ATI, you are going to read about lysosomes. And whenever I see lysosomes, I always think of a can of Lysol. Students, you've probably seen cans of Lysol. Maybe you've seen them at home or at the store. It is a disinfectant. It kills germs. So think of your lysosome and Lysol as kind of being the same thing. You also have something very similar called peroxisomes that breaks things down. Think of it like peroxide, and it's breaking down fatty acids. The cell is not Las Vegas, so what happens in the cell does not stay in the cell. Things move to and from. Sometimes energy is needed, sometimes no energy is needed. So let's talk about transport. So for starters, I want you to understand that human cells or eukaryotic cells have a cell wall, and that cell wall is made of a lipid bilayer. So we've got our cytoplasm in here, that's in the cell, your extracellular fluid that's outside of the cell. Here is your cell wall. I'm not very good at art, but just bear with me. So what is your bilipid layer? You have a hydrophilic head, and anytime you see philic, think loving. And I know this is a pretty awful comparison, but this is how I keep it in my brain. A pedophile is literally means lover of children. So if when you see philic or phile, that means loving. So in the case of your bilipid layer, a hydrophilic head means a water loving head. So that is the little pink circles right here. That is the water loving side. And so that is the side that faces the fluid, that faces the water. It also has a hydrophobic tail. Hydro means water, phobic means fearful. So like arachnophobia, which is a fear of spiders, which I have. I don't like them. Think of how oil and water don't mix. So your hydrophobic tails means that they don't like water. So we've got our hydrophilic heads, so that points towards the water, our hydrophobic tails, and they try to stay away from the water as much as possible. Okay, so things still get through the cell wall. It doesn't keep everything on the inside and everything on the outside, but how things move in between is usually through proteins, carbohydrates, or cholesterol, or just simple effusion. So sometimes that transport does not need energy. So passive transport, this guy is getting transported. He is putting no energy into it whatsoever. And ways that we can transport things in and out of the cell wall is without energy is simple diffusion, facilitated diffusion, and osmosis. So passive transport means he's just a passenger and he's not using any effort to get from point A to point B. Active transport. So active transport needs energy, which is our ATP. So a great example of that, going back to anatomy and physiology, the sodium potassium pump exchanges out 3 sodium ions for 2 potassium ions. So you don't need to remember this right now. Just remember you have a sodium potassium pump. So this is our little pump. Here is our sodium, here is our potassium, and I plugged it in because ATP is energy. So ATP is energy. It's pumping sodium and potassium, and it needs energy in and out of the cell wall. Okay. Another thing that needs energy and kind of moves things is pinocytosis and phagocytosis. So, pinocytosis is when like the cell kind of drinks something up, and I think of Pinot Grigio, which is a type of wine. And phagocytosis, phago means pig or to eat. So that is when the cell eats something. So here's our little phagocytosis. Exocytosis is moving proteins out of the cell or secreting hormones. So here is our little cell, and it is secreting hormones. And I just made it look pretty. So exo, think exit, eviction. So exocytosis is exiting or evicting something out of the cell. So what is the fuel? Mostly the mitochondria uses glucose. It can also use fatty acids and amino acids, but our primary source of energy is glucose. And remember, the mitochondria is the powerhouse of the cell with its maternal only DNA, and it makes ATP. So glycolysis. Now that's the breakdown of glucose. Remember, we just said that glucose is the primary fuel. So glycolysis is the breakdown of glucose. It only makes 2 ATP molecules. It also makes some pyruvate, and it needs enzymes to make this happen. So think of this graphic that I've made you guys. So here we have our glucose. It's a lovely little marshmallow. We've got our enzymes down here that's cooking something. And as the result, we get 2 ATP and some pyruvate. Now, if we add oxygen to that, so I've got our little O2 down here and we've got their fan blowing on it. Oof, now we're really cooking. We've got our glucose, we've added oxygen, and oh my gosh, look at that. Now we're making 32 to 34 ATP. And at the cellular level, when we're thinking of all the cells in our body, oh my gosh, we need oxygen to make energy. Okay, this is the best. So what happens when oxygen levels are insufficient? Pyruvate converts to lactate, and that regenerates to make more NAD needed for continued glycolysis. So it's a coenzyme needed to break down glucose. So if we look over here and we don't have any oxygen, and we keep not having oxygen, we've got our pyruvate that turns into lactate. Okay, lactate. That's not good. We can draw a blood level called the lactic acid level. Lactate, students, lactate is a sign something bad is going on with your patient. So high lactate levels in the body, also known as lactic acidosis, are concerning because they are telling you the cells are not getting enough oxygen to efficiently produce the energy for a thorough uh, aer- aerob- metabolism. Apparently, words are hard right now. I apologize. Instead, the body shifts to anaerobic metabolism, which produces lactate as a byproduct. Here are your two conditions that I want you to think of when you don't have enough oxygen and it's causing an increase in the lactic acid level. So sepsis and tissue hypoxia. When your patient doesn't have oxygen and they start going into anaerobic metabolism, they get a high lactic acid, and these are the two things that I want you to think of. How do you keep it separate in your mind? So aerobic respiration, well, you need oxygen to work out and do stuff. Okay, that makes sense. Anaerobic means you don't have oxygen. You're still doing a little something, usually not up to any good, and you don't have air. So not breathing O2, but still doing something. Remember, students, anytime you stick the prefix AN in front of something, it means without. So this literally means without oxygen. This is with oxygen. This is without. Let's go on to DNA. All right, DNA. What is DNA? Think of it as the blueprint. You've got 23 pairs. It's a double helix. So see, we've got one helix, you've got the other helix, and you got 23 pairs of them in you. You've got these nucleotides. There's 4 types. That's what these different things are. There's 4 different nucleotypes. DNA. It is functional. It has sections on it that create specific proteins and also regulatory RNA molecules. It contains your genetic information. The gene expression goes through transcription, so it uses messenger RNA, and translation. It makes the protein. RNA, nucleic acid. Think of it as the runner. RNA is the molecule found in living cells that plays a key role in making proteins and carrying genetic information. Think of it as a messenger. It reads instructions stored in the DNA and then carries them to the part of the cell that builds the proteins. And students, do you remember back up here, what is the part that builds the proteins? Your rough endoplasmic reticulum with the ribosome studs makes your protein chains. Come back down here. Okay. 3 main types of RNA. We've got messenger RNA, which carries the genetic instructions from the DNA out to the ribosome. So he's the messenger. Transfer RNA, he brings the correct amino acids to build the protein, and then ribosomal RNA makes up the ribosome. It is the cell's protein building machine. Just a quick comparison between RNA and DNA. Um, we've got DNA, it's deoxyribose, RNA is ribose. Strands. DNA is double-stranded, our RNA is single-stranded. The bases or the nucleotides are very similar, but we have a T over here on the DNA and a U on the RNA. And location, RNA kind of goes all over the place. It goes in and out of the nucleus, the messenger RNA, and then the cytoplasma. So here's a little graphic I made. So we've got our messenger RNA. He's got the instructions off the DNA, and then we've got our transfer uh, RNA. So he's going to go run and get the right amino acids, and then they're going to bring them over to the ribosome builder, and he's going to make our protein chains. Now, it doesn't always, when the messenger RNA goes into the nucleus, he doesn't take a whole copy of like the DNA. He just takes a section of the DNA because DNA is complex and it's got a lot of information on it. So of the whole strain, let me come back up here. So of the whole strain, the messenger RNA might start to copy here from a start codon and maybe to here. And maybe just this little section has the information of what it wants to build for that day. So it has a start codon and a stop codon, and there's our uh, newly synthesized protein. So RNA, a nucleic acid essential for all living cells that carries information from the DNA and controls protein synthesis. Our ribosome is a protein building factory. Think of it as a machine on an assembly line. So our mRNA, our messenger RNA, carries genetic instructions from DNA. The ribosome reads the mRNA code, 3 letters at a time, called a codon. The tRNA delivers the matching amino acid for each codon. The ribosome links the amino acids together, and then you've got your protein. So think the difference, the cellular machinery actually reads the signal to begin or end the protein, that's mRNA. And if you are looking at the blueprint itself, that's DNA. So what could possibly go wrong? Well, there could be a transcription error, or in my case, a spelling error. I forgot that N. Protein folding gone wrong, or genetic mutations. This whole process, students, is really complex. So if the mRNA accidentally reads one wrong, then it's going to transcribe the protein wrong, and then we're going to have other issues. Or it might fold the protein wrong. So instead of the protein looking like this, it might fold it incorrectly, and then your body looks at it and misreads it. Or a genetic mutation. Maybe when the DNA was replicating, it accidentally had a little whoopsie and has a little mistake here. And now every time that messenger RNA reads it, it reads it wrong and then makes a wrong protein, and it just gets bigger from there. Some examples of inherited genetic diseases. So we've got Down syndrome. Hypertension can be genetic, Fragile X syndrome, some cancers like breast or prostate cancer. If inherited, there's the Mendelian patterns. You guys probably learned that in anatomy and physiology. So autosomal dominant, you just need one parent to get it. Autosomal recessive, you need two parents. So just think of dominant. If it's a dominant inheritance pattern, obviously it's going to be more, it's going to erupt more. If you just have one parent, you got a 1 in 2 chance of getting it. Some examples of Huntington's chorea, neurofibromatosis, and high cholesterol. And then recessive, you basically got like a 1 in 4 chance. If only one parent has it, 50% chance if two parents have it. And you guys have probably seen these genetic inheritance patterns before. Epigenetic regulations. So epigenetic regulation, let's kind of go into some examples. So that is changes in a gene activity that does not involve alterations to underlying DNA sequence. Instead, these changes affect how genes are turned on or turned off, essentially controlling gene expression through chemical modifications to DNA or its associated patterns. So what does that mean? Um, with development, it guides cells and how stem cells become specialized, response to environment, diet, stress, toxins, disease. So if you get certain diseases, it will change how your body reacts from here on out. And inheritance, sometimes you can inherit things. All right, so let me come down here to this example. Oh, no. Okay, we're going to kind of change gears and kind of talk about stem cells here. Stem cells, what are stem cells? Well, they self-renew. They keep regenerating, and they can keep regenerating over long periods of time. The nice thing about stem cells is they can differentiate into different types of specialized cells. Oops. Why is this not loading? So let me see if I can move this over. Okay. Oh, there it popped up. Okay. So embryonic stem cells, these are found in embryos, can become almost any cell type in the body. So pluripotent. Adult somatic stem cells. So these are found in tissues like bone marrow, skin, and the gut. can usually only become a limited can become a limited amount of cell types. So multipotent and induced pluripotent stem cells. This is outside of the scope of this class, so don't worry about this. If you ever take advanced pathophysiology, worry about this. For the purpose of this class, I only want you to worry about embryonic stem cells and adult somatic cells. So in embryonic stem cells, once upon a time you have an egg and a sperm and they get together. And when that happens, you get a fertilized egg and then it divides and divides and divides again. And that's all it does is divide and divide and divide. And it takes about 2 weeks before any differentiation happens. So this cell division is super, super fast. So this is an embryonic stem cell, meaning that these cells can then grow up to be absolutely anything. Once you get past 2 weeks, that stem cell starts to differentiate. You have three main germ layers. You have your endoderm, your mesoderm, and your ectoderm. Your endoderm is the lining of the GI tract, stomach, respiratory stuff, liver, pancreas. Your mesoderm, or your middle uh, differential layer, muscles, bone, cartilage, kidneys, heart. And the outer layer is your ectoderm. So that's where you get stuff like skin, nervous system. And we'll go over this more as we go through the class. Once you start having differentiation, cell division isn't as fast. So this, when it's just an embryonic stem cell, it's not thinking about it. It's all it's doing is division. And those cells can become anything. Once you have differentiation, so this would be like your somatic embryo, your somatic cells. They have some differentiation, but they can still become different things, but they're a little bit different. So adult somatic cells, that's this. You've got your hematopoietic stem cells from the mesoderm. Those generate all blood cell types, red, white, platelets. Your neural stem cells from the ectoderm, this guy right here, generates neurons, astrocytes, oligodendro, I can't say that. Words are hard. And then you've got your intestinal stem cells from the endoderm that make your stomach cells, goblet cells, endocrine cells. All right, so now that you have all of these cells, they talk to each other. So there is something in your body called cell signaling. So we've got these two cells and they're chatting. We've got good old-fashioned landlines here and they're chatting with each other. So we've got, oops. So the four main types of cell signaling, we've got autocrine signaling. So that's when the cell tells itself, hey, we need to step it up or we need to do something else. Paracrine signaling is when that cell is communicating only with other cells that are like it. So that's para. So para signaling, only other cells that are like it. Synaptic signaling, that's between two neurons. So just kind of jumping over a synaptic cleft. And then endocrine signaling, that has to do with hormones and cholesterol, and that can communicate over long distances. So over your bloodstream. So that goes into your bloodstream and goes other places. Cellular adaptation mechanisms. So we've got four main types. We've got atrophy, hypertrophy, hyperplasia, metaplasia, and dysplasia. Burn this into your mind. So we've got cellular adaptation changes. So here we have a normal cell. This is atrophy. That is where the cell gets smaller. Hypertrophy, that's where our normal cell still looks normal but gets bigger. Hyperplasia means it's multiplied. So we have this, but now we got a whole lot more of them. Metaplasia, oh, this isn't good, guys, because now we had our normal cell, but now it's changing how it looks. And then this one is the most scary and sad. This is dysplasia. So now we had our normal cell, but then now it's turned into like in a regular shape. It's not really recognizable. Maybe you can tell it was originally this type of cell, but this doesn't look right. So with atrophy, the size of your cell becomes decreased. The number of cells can become decreased. Sometimes it can be reversed. Examples are muscle disuse. Hypertrophy, size of the cell is increased. Usually you have the same number of cells. It is sometimes reversible. And things would be like cardiac muscle, skeletal muscle, and prostate. Hyperplasia, uh, the number is, or the size remains unchanged, but now you have more of them, like liver regeneration. Metaplasia, the size of the cell may get bigger, may be smaller, may stay the same, same with the number. This is harder to reverse, and that would be like respiratory and esophageal changes. And dysplasia, this is the scariest because this is the most extreme adaptation or change to a stress. The size is variable, and so is the number. So let's go into examples because that just makes things make more sense when we see examples. So an example of atrophy of a cell. Think of your athletes who are maybe paralyzed from the waist down. So those muscles now are no longer getting used and so they shrink. That is a normal cellular response. They're not getting used anymore and now they have atrophied or they have shrunk and gotten smaller. That can also be a normal part of aging, like your kidney, your liver, and your skin. An example of hypertrophy would be hypertrophy of the prostate. Girls, we don't have a prostate gland. Only boys have a prostate gland. So here's the bladder. Here's the prostate gland that we find in men. That is a gland that is right below the bladder and goes around the urethra. And this is a normal size. Sometimes it gets enlarged and it just does. And that can cause some urinary problem, but this is an enlarged prostate, and that is an example of hypertrophy. Another example of hypertrophy is working out. So you're making these muscles work harder and harder, and in response, they get bigger. Another example of hypertrophy, here you have a normal heart, and when your blood pressure is going up because of other vascular changes in the body, your heart responds. And so it starts to get bigger and bigger. And in the world of cardiology, a big heart is not exactly a good thing. Look at it on the inside. So here's our normal heart, and here's our hypertrophic heart. So see how the heart muscle got bigger? Our chambers got smaller. So now it's not pumping blood as well. It's not very effective. So when you make the heart work harder, the muscle responds and gets bigger. When you make your skeletal muscles work harder, they respond and they get bigger. But sometimes it just happens as a normal part of aging. Hyperplasia, the liver can regenerate. So sometimes if you have an insult to the liver can regenerate and make more liver cells. More. Normal uh, puberty process with women is a great example of both hypertrophy and hyperplasia, both with breast tissue. Not only is with normal endocrine changes or puberty changes with um, women, you get not only larger breasts, but you get more breast tissue. All right, so metaplasia, cells that were there before change to something else. I got my frowny face. So here's an example of Barrett's esophagus. So this is a stomach, and it is normal, students, this makes sense, normal to have cells in the stomach that make mucus because we have acid in our stomach. So the stomach wants to protect itself. So it has these cells that secrete acid and it protects the lining of the stomach. And then in your esophagus, students, we have that is lined with squamous cells, and that is normal. Okay. Well, sometimes if somebody has really bad heartburn, and they have really bad heartburn for years and years and years and years, and it starts burning the lower part of their esophagus. Well, their body tries to protect itself, and so it undergoes cellular changes. So the cells that used to be squamous cells here at the bottom of the esophagus change to columnar cells. Now this is scary, students. So the body's trying to protect itself. Now it's putting columnar cells there so it can secrete mucus and protect the lower lining of the esophagus from all that acid. But anytime you change cells from one thing to another that weren't there before, this is a higher risk of cancer. So patients who have Barrett's esophagus have a higher risk of esophageal cancer. Here's another example. People who smoke, okay, or people who breathe in chemicals in their lungs for years and years, and that's a bigger risk. Normally, the lining of your bronchioles is made with something called ciliated columnar epithelium. So these cells right here, we've got the cilia, and that helps, like, get out pollen and garbage and stuff like that. But when you have years and years and years of smoking and putting smoke over this, oh gosh, well, your lungs don't like that. So it tries to protect itself. It undergoes cellular changes, and it starts making squamous cells. So instead of having normal columnar cells, it actually makes the cells inside their lungs different. And then they have to cough a lot more because now they don't have all of these ciliated cells. So they get what we call a smoker's cough because they've got squamous cells lining their bronchioles now instead of columnar epithelial cells. Okay. If you ever know anybody that quits smoking, they will tell you the first 3 weeks are usually the worst as far as how they feel in their cough. And it's because their body is trying to go back to the way it was prior to smoking. So they're coughing all this stuff up, and the body's trying to fix itself. But this is the why behind it. So in our bronchi, so we've got our ciliated cells that we're supposed to have, but in smokers, not so much. Our goblet cells, which normally produce some mucus, but our smokers have a lot more because it's trying to get that tar and that nicotine and that smoke and all that other stuff out. Your basal cells, those are normal, but now it's abnormal in smokers. They've got increased proliferation, or it's making more. Squamous cells, we normally don't have those in the bronchi, but when you have metaplasia with our smokers, they do. Dysplasia. This is the worst of all of your cellular changing and responses. So loss of uniformity disturbs cellular growth and differentiation, and it can progress more easily to malignancy. Once you start having dysplasia, that is a much higher risk for cancer. So cellular dysplasia example. So tanning, freckles, mole changes, cancer. So if you have somebody who frequently gets sunburns and they tan a lot, they're forcing their skin to make cellular changes. And you keep doing that over and over again, and then you're going to be at a higher risk for having things reproduce wrong and it turn into cancer. So normal squamous cells become atypical. The cells may show enlarged, irregular nuclei, increased cell division, loss of normal cell organization. They may appear immature or abnormal. In advanced stages, this can become cancerous. All right, so now we are going to talk about the general adaptation syndrome. So, what the general adaptation syndrome is this hypothalamic pituitary adrenal axis. And your Engage series is going to go into this in depth. That's a lot of work or words. So, we call it the GAS, the general adaptation syndrome. It's in 3 stages: alarm, resistance, exhaustion. So, let's talk about it. So, alarm, that is where your body sends the alarm. It notices something is wrong and it pulls the alarm, shouts at everybody, and says, "Hey, we have got to mobilize our resources. A stressor just happened. Call for help." Then it goes into the resistance stage. So your body makes some changes and it's like, all right, I've got this. I'm going to build up this resistance and deal with this stressor. However, coping doesn't always happen, and sometimes you get to the point of exhaustion. All your reserves are depleted. So here's a nice hormonal graphic example of this. So our hormonal pathway, stress and salt. So you've got your hypothalamus. It then signals the adrenal gland, which is above the kidney. As students, if you haven't figured it out, the word renal means kidney. The word adrenal means adding it to the renal, so adding it to your kidney. And then your adrenal gland makes cortisol, which is your stress response hormone. And then it also affects aldosterone, which is fluid balance. But we are mainly talking about the hypothalamus, adrenal gland, and your cortisol response today. So chemically, what happens? Your hypothalamus releases corticotropin releasing hormone first, which corticotropin releasing hormone stimulates the pituitary gland to then release adrenocorticotropic hormone. And then that increased circulating ACTH triggers the adrenal glands to increase production of an intrinsic glucocorticoid such as cortisol and aldosterone. So I kind of like this little graphic that I made instead. This is just easier. Bink, bink, bink. Here's the big words. Okay. All right. Our next section of the cell that we're going to talk about is apoptosis. Now, I have heard it pronounced both ways, apoptosis and apoptosis. I will pronounce it both ways. Neither one's wrong as far as I'm concerned. All of your cells in your body are supposed to have a pre-programmed death. So in with the new, out with the old, out with the old. So here's our red blood cell, and he is supposed to expire at 109 days. And this is kind of your body's way of cleaning stuff up and, uh, in with the new and out with the old, because we don't always want to have like the same cells trying to do the same thing day in, day out, because they get old. So your body tries to regenerate a lot of stuff. So like your red blood cells, they carry iron, they carry oxygen. And after 109 days, your spleen and your liver kicks out the old one while your bone is making new red blood cells, for example. In cancer, if you've ever seen people with cancer, they look very emaciated, very skinny. Cancer is smart and cancer is hungry. And this is what I mean by that. Cancer often evades normal apoptosis, which is a crucial process that removes damaged or abnormal cells from the body. So instead of cancer cells dying with apoptosis, they keep living. So now we've got more cancer cells, and they also cancer is smart. Cancer Cancer will not only not kill itself off like it's supposed to with apoptosis, it tells other healthy cells to feed it. So now it's taking nutrients from other healthy tissues to feed it. So it's eating more, it's taking more resources, and it's not killing itself off. That is why a lot of cancer patients look very emaciated, because that tumor is taking resources, and also that it's not undergoing apoptosis. So you got this little cancer cell here. I refuse to die. It tricks other cells around it into feeding it, triggers new blood vessel growth to feed it, and ignores normal apoptosis. And we're going to go over cancer more later in the semester. And actually, one of our drugs that we use to treat cancer actually targets the blood vessels that feed cancer, so that it doesn't get as much nutrients. All right, next section, cellular response to injury. So a cellular response, one of them is necrosis. So that would be cell death from ischemia, toxins, and trauma. Ischemia example 1. So here's our normal heart. Here is our heart that's had a heart attack. So now when you have a heart attack, there is blood flow that gets blocked off to this section of the heart. It becomes necrotic, and then your body cleans that up, and then it replaces it with just connective tissue or scar tissue. So once a section of heart muscle dies, you don't get that back. So we really don't like people to have a heart attack because then it's no longer cardiac muscle. It just gets filled in with connective scar tissue. Ischemia example two. So if you have blood flow that gets cut off to your toe, it's going to turn black and die, and it'll eventually fall off. It'll eventually fall off, and that's if it's dry gangrene. If it can also get infected. A lot of times this is infected. Botulism toxin, it blocks nerve signals leading to paralysis and cell death. Yes, Botox is derived from this and it's purified. So a lot of us like our Botox. I haven't had any yet, but maybe for my 50th birthday. Trauma. So we've got actually taking off, doing an amputation. Obviously, these cells are going to die because they are no longer getting nutrients. So trauma. Anytime you lose a limb, that's going to die. Okay. So that is that portion of it. And we have gone through 90 slides. So I am going to stop this lecture here and I will have the second half uploaded. So let me know if you have any questions. Thank you.